All right. Well, you've all heard a lot this morning and this afternoon about the intersections across FTD and related conditions. I just want to start off with a quick reminder that it's really complex. I was very impressed with Dr. Holden's slide about people's minds almost exploding during their clinical training, right? So we have clinical overlap with Parkinson's, Alzheimer's disease, ALS, psychiatry. We also have biological overlap, and the focus of this conversation now is going to be diving in more on the biological aspects of how do we accelerate the pace of research, both so that it can improve clinical care and so that we can get to the newer treatments and the better diagnostics that will help improve families' lives. So I'm joined today by, as Esther Keene outlined, Dr. Michael Benatar. He is professor of neurology, the Walter Bradley Chair. This is a long list, Michael. So.
Be brief.
The Walter Bradley Chair in ALS Research, executive director of the ALS Center, chief of neuromuscular division, and vice chair for clinical and translational research in the Department of Neurology at the University of Miami. And very specifically, he leads an active clinical and translational program to develop biomarkers and therapies for ALS. He is the principal investigator of an ongoing pre-symptomatic familial ALS study and the CREATE Consortium. And he has been a real, we often call you an honorary member of the FTD field because of how much your work has really been proactively collaborating with the FTD space. And Dr. Corey McMillan is an associate professor of neurology at the University of Pennsylvania and co-director of the Penn Frontotemporal Degeneration Center. And Dr. McMillan's laboratory is working to develop biomarkers to help diagnose neurodegenerative diseases, accelerate drug discovery of disease-modifying agents, and to define essential elements of clinical trial endpoints. And again, specifically for this session, Dr. McMillan has been a leader in the collaboration from FTD to ALS. And so we're really, really grateful that both of you can be here today to talk through some of these different pieces together. I think we want to start off with just the basics. What is the relationship between FTD and ALS? Dr. McMillan, you want to go first?
Sure. Maybe I can comment a little bit on the biology, and then Michael can talk a little bit about the clinical overlap. So a theme that you'll hear about in this session is the importance of cross-disease studies. When we started banking brains at the University of Pennsylvania about 30 or 40 years ago, the clinicians doing this recognized the importance of banking brains and blood and collecting the same clinical data across our FTD clinics, our ALS clinics, our Alzheimer's disease clinics, and our Parkinson's disease clinics. And by doing that, we were able to identify commonalities and shared features of disease. In 2005, only half of the brains in the brain bank stained for a specific pathology, which is tau. And the other half of FTD brains, we didn't know what the protein was. And in 99% of ALS brains, we didn't know what the protein was. The next year, TDP43 was discovered in FTD, and clinicians who had been looking across diseases recognized that some people had FTD, ALS, or a combination of both within families. And through that, they also identified that that same protein, TDP43, that was part of FTD, is also was then accounted for the other 99% of ALS brains. So this provided a real strong biological link between FTD and ALS. 5 years later, C9orf72 repeat expansion was identified, which is the most common genetic cause of both FTD and ALS. And we know that even within families, brothers and sisters, one can have FTD, one can have ALS, or they can have combinations of both. And then as we learn even more, we know that there are other types of genetic variants that relate to the way that genes are expressed, or smaller components of the genome that also modify the expression of a cognitive or a neuromuscular syndrome. So there's an increasing recognition of the biological overlap between the two. If you want to comment clinically.
Yeah. So perhaps just to add, in addition to this biological overlap and the genetic overlap, there's also a clinical or a phenotypic overlap. So people with ALS can develop FTD. They can also develop milder versions of FTD, what we call cognitive impairment or behavioral impairment of the frontotemporal variety. So sort of a form thrust or a milder version of an FTD syndrome. Similarly, patients with FTD can develop ALS or a milder version of that where there's some motor manifestations that are along the spectrum of what we see in ALS. And so what we increasingly recognize is that clinically or phenotypically, these two diseases exist along a spectrum, where some people have pure ALS, some have pure FTD, and then some with different combinations of those two in between. And so I think the clinical line of evidence, the genetic line of evidence, the biological line of evidence, at some level tells us these are very much related diseases, if not the same disease, and I use the word disease there from a biological perspective, with ALS and FTD being two different clinical or phenotypic manifestations of what that underlying disease is.
Yeah. So one of the challenges we encounter is the inherent complexity of both FTD and ALS. So on the FTD side, not all cases of ALS are on that spectrum like you're describing, right? And I think there's a similar type of complexity within the ALS space about its overlap with other types of conditions that are not FTD, for example. There's such a challenge to overcome here that one thing that really struck us a few years back was AFTD was hosting a Holloway Summit for digital assessment tools. But how do we measure people's function and abilities within the comfort of their home so that we could then use that as a research tool that then requires less travel when someone's participating in research? And Dr. Benatar participated in that meeting. And it was so striking to hear a lot of speakers from the ALS space talk about their cognitive tools that they were developing, completely independent from the tools that the FTD field was developing for the same measures. So that was where, that led where AFTD eventually partnered with the ALS Association, and we did a funding program specifically to try to help align these. But it's so complex. So I just wonder, could you talk a little bit about why are these types of silos here, and how do we, what kinds of challenges do they create or pose for the progress in the research space?
Yeah, so I can comment that I think, as Michael commented on, is that there are unique clinical issues associated with the treatment and clinical management and care of an individual with neuromuscular syndrome and an individual with a cognitive syndrome. So it makes sense that there is some extent of siloing. But there also is an underappreciation of the overlap between the two disorders in clinic. So typically in an ALS clinic, people aren't evaluated for cognitive disorders. Typically in a cognitive clinic, people aren't evaluated with an EMG to understand whether they have underlying neuromuscular disease or at risk for neuromuscular disease. Some of the challenges are associated with the practicalities of assessing these things in clinic. So many neuropsychological tests or cognitive tests have motor components. So you have to fold a piece of paper, or you have to speak or hold a pen. And in ALS, your motor impairments can confound your ability to do those things. So you might score poorly on a cognitive test, not because you can't think through the questions, but because you can't hold the pen to answer the question appropriately. So there's been a lot of work in the ALS field now to develop tests that can account for this motor impairment. And that has been really important. But there hasn't been normative data, and Michael and I have been leading an initiative to better understand what the cutoffs are of cognitive impairment in ALS and how to take our cognitive knowledge from FTD to apply to interpreting ALS test results.
Thank you. And before we go to Dr. Benatar, Dr. McMillan, you published a paper recently about the prevalence, particularly in C9orf72, of FTD, ALS, or a combination thereof. And to me, this is a real example about just how challenging the space can be, because if people are being diagnosed in ALS clinics that are not recognizing the presence of FTD, and this opposite is happening to some extent in the FTD clinics, it becomes a challenge. If you're a given family member how do you know what your future risk might be if you have a given gene, or particularly C9orf72, in your family? Can you talk about that recent study?
Sure. This is a question that comes up in clinic all of the time, where an FTD patient wants to know what's their risk of getting ALS, or vice versa. And the question is difficult to answer because historically, cognitive information wasn't assessed in ALS clinics and motor information wasn't assessed in FTD clinics. But through our shared brain bank of having 30 years of brains across both FTD and ALS, we were able to look at the natural history of hundreds of patients and understand what the risk is of, and the what we call time to events occurrence of developing both symptoms. And what we found is that individuals with a C9orf expansion have a 4 times increased risk of developing both FTD and ALS. And that risk cumulatively increases over time, over the course of the disease course, in a pretty linear sort of way. About 10% of individuals that developed FTD and ALS did not have a C9orf72 expansion. So C9 expansion doesn't explain all of it. And we're looking into other genes and other genetic factors that might explain some of that increased risk. Of those that don't have a C9orf expansion, what we find is that the co-occurrence of FTD and ALS plateaus after about 3 years of disease, so that the risk is higher at first, but then after about 3 years, it plateaus and the risk of developing both is unlikely. And then finally, the risk of developing both FTD and ALS does not differ whether an individual starts with FTD or whether somebody starts with ALS.
It's interesting.
Yeah, I just wanted to add a word about sort of why we find ourselves in the position that we're in with these silos. And part of this is historical, a big part of it. And so if you read the ALS textbooks, until very recently, they talk about weakness and motor manifestations of disease, and they'll go so far as to say that cognition is preserved. Right. And so this is how we're trained. We're trained as neuromuscular doctors to look after and to identify ALS. The cognitive neurologists are trained in cognition and behavior and not in neuromuscular disease. And so to use the old adage, to a man with a hammer, everything is a nail. You see what you're looking for as opposed to what's there. And all credit to the people and the groups who mapped and identified the C9orf locus. And a key part of that was recognizing that ALS and FTD coexist in the same families, are part of the same underlying disease. And maybe to say, as we now recognize we're seeing our blind spots in the rearview mirror, I think we have other blind spots that we're still not seeing. ALS and FTD aren't the only silos. Mm-hmm. There are other genetic causes of disease, for example, VCP, where there are also muscle manifestations of disease or bone manifestations of disease, even extrapyramidal or movement disorder, basal ganglia diseases. And we still remain very siloed in those other areas, and we need to, as we're breaking down the silos between ALS and FTD, think about how do we learn from those sort of historical misjudgments or mistakes to do better going forward across the full spectrum of manifestations that we encounter.
So, Dr. Benatar, there was a very specific workshop that you co-led in the last year that was around prevention of disease in C9orf72, but more specifically, how might a clinical trial be designed to prevent disease? And I thought it was just so striking in how effective that was, bringing in ALS leaders and FTD leaders who are largely independently creating the tools that might be used in those trials. So how do we anticipate what those pieces might be? Can you talk more about that workshop?
Yeah, so for those who don't know, I've had a long-standing interest in the idea that it might be possible to prevent ALS. And as I've matured in my thinking, it's not just ALS, but FTD. And C9orf72 provides a unique example. If we are looking at an unaffected carrier, and we're thinking about a prevention strategy that we want to intervene therapeutically before people develop a clinical manifestation of disease, it's clear that we have to prevent both ALS and FTD. We can't have the ALS community thinking, how do we prevent ALS, and the FTD community thinking, how do we prevent FTD? We need to prevent both of those manifestations. So that takes us back to thinking about biology, and it really forces these two communities to say, how do we overcome some of these obstacles? We're each doing preparatory work, but if we're really going to get there, we have to work together to make sure that we have the tools in hand and our thinking is congruent and concordant with each other, that when we get a chance to do disease prevention, we're going to have the best shot at preventing all of those phenotypic manifestations. And it was an enormous credit to AFTD and to the ALS Association to come together to work with me and Dr. Boxer from UCSF to say, how do we pull all of the relevant segments of our community together to make sure that we are laying the necessary foundation to be able to prevent disease? Again, I forget who said it this morning, but we're going to be successful if we work on these problems together. We're not going to be successful or not nearly as successful if we work separately. So prevention is a perfect example of how we have to work together.
I think it's also a perfect example of where hearing from families can also drive the questions being asked. Because when you ask an ALS research group what is their interest, it's natural that it will say, we want to figure out who's going to develop ALS so that we can prevent disease. We want to prevent ALS. But when you speak with the family, and we've had volunteers speak to the experience of having identical twins. One develops FTD with a bit of ALS, the other develops ALS with a bit of FTD, and how in those settings, those family members don't want to just have prevention of ALS. They want to have prevention of C9 disease. And to your point about other types of genetic risks as well, it's prevention of disease, period. And we were really proud that there were folks from both a deeper FTD background as well as a deeper ALS background who could be representing that lived experience in those meetings. And that is what we consistently hear from the families that we support. One of the big challenges specifically in that issue is that even on the clinical care side, genetic testing is extremely different. Your experience as a family will vary depending on which clinic you walk into. Would one of you be able to talk about that difference between the FTD and ALS spaces? I'll leave it to you and who.
I think it's critical to have well-informed genetics counselors that are aware of the equal risk of FTD and ALS. Across specific clinics, across diseases, the frequency and implementation of routine genetic counseling has varied widely. In ALS, I think it's becoming increasingly more common that everyone receives genetic testing as part of clinical care, or at least in specialized centers, that's the case. In FTD, we've been slow in order to adopt that, I think. And it's unclear why that's the reason. And I think families, like all of you here, speaking up about the importance of genetic testing and letting people know that you want it will be really important. We know that family history is the strongest predictor of genetic risk, but we do find genetic causes of disease in sporadic families that don't have a family history as well. So it's an important clue to understanding each individual's disease.
Maybe just to add, this is another opportunity where our two communities can learn from each other and benefit from the experience that's developing, evolving in another community. And I'm often struck by what I understand is very low levels of genetic testing in the FTD community, but increasingly very high levels of genetic testing in the ALS community. I think the trigger for us as an ALS community, I think there was already a broad recognition that we needed to and wanted to do genetic testing. Like in FTD, we find genetic causes of disease even when there isn't a family history of disease. That's critical that we don't equate familial with genetic. As Dr. McMillan has said, when there's a family history, higher likelihood of finding a gene, but we also find genetic causes when there's no family history. And the catalyst for us was the emergence of sponsored testing from our industry partners, to make testing easy, because we used to struggle to get commercial approval for this. I think the additional catalyst for us in the ALS community is that we now have an antisense oligonucleotide or gene therapy for people with SOD1 ALS. That's only 2% of all ALS, but this is a very meaningful therapy, especially when initiated early. And so now we are on the hunt, so to speak, to find anybody who carries an SOD1 variant for whom we can now offer them a meaningful therapy. But the next step is getting into disease prevention. And so I'm thinking about cascade testing here, and why this is so important for the FTD community too, is that finding the genetic cause in an affected individual is the first step to them being able to offer predictive testing to family members. And that's empowering from a research participation perspective, but also as we're gearing up and preparing for disease prevention trials. And again, if we're going to be successful in prevention, we need to find families and carriers from both our FTD and our ALS clinics.
So there was a lot of information in both of your answers. I want to reinforce one piece, that in order for research to collaborate across the two spaces, we have to know who has C9 or F72 across those different clinics. And that is, I think, one of the bigger challenges with certainly in the FTD field, to collaborate with the ALS field on some of those pieces. And Dr. Benatar, you also flagged the prevention trials a couple moments ago. And you've been very generous of coming to our FTD Research Roundtable, where we're talking about clinical trial design with the different companies focused on FTD and the different regulators and the academic research leaders and lived experience representatives, to talk about your experience in prevention trials. And I know that almost every family member will say that that's what we want. We want to prevent it from happening. Can you talk a little bit more about what it takes to have a prevention trial, and about some of your experience in the ALS space with that?
Yeah, so it's a great question. And what Penny's referring to, if this community is not aware, is we have actually started the first prevention trial in the ALS space. And so we have been studying for almost 20 years through the pre-FAL study, the pre-symptomatic familial ALS, unaffected carriers at elevated genetic risk. And the first gene we had was the SOD1 gene, and so that formed the basis of a lot of the things that we had learned about predicting the emergence of ALS as a clinical syndrome. And one of the things we found is that this protein, neurofilament light, abbreviated to NFL, different from the NFL most of you are probably familiar with. Although I will say head injury in the NFL leads to a rise in NFL, but that's a topic maybe for another discussion. But what we found is that this protein, NFL or neurofilament light, goes up in the blood 6 to 12 months before SOD1 carriers develop ALS. And so we made that discovery just as Biogen and Ionis were bringing their SOD1 antisense oligonucleotide into the clinic and finding initial proof of concept that this was going to be safe and perhaps beneficial. And so that led us to go to Biogen and to say, could we take this information? We now have a biomarker that tells us when disease is coming. Could we use this to identify people in whom we could offer them this gene therapy before development of disease, to try to prevent disease? And that trial, the ATLAS trial, is now ongoing. And we're learning very important things that will be valuable to the design of prevention trials in the C9 population, I'm sure in the granular and the MAPTau populations. But just to underscore that they're not identical. So I don't think that NFL alone is going to be sufficient in, for example, a C9 population. We're going to need other biomarkers as well to help us identify who are the people in whom overt disease, ALS or FTD, is coming soon. And Dr. McMillan's been doing some fantastic work on the imaging front and other biomarkers that we might be able to combine these to sort of tell us who can most benefit. And the key thing, sorry Penny, short question, long answer, is that we don't want to wait until we have a therapy that we want to test preventatively, to think about the design of a prevention trial. We want to be doing all of that foundational work so that by the time we have a therapy that we think has a good shot at preventing disease, we are ready to go. And that's how we're going to compel our industry partners to say, here's all the foundational work. We are ready. Work with us to take your drug or your gene therapy into this population. And it's how compelling our data are and how ready we are that's going to make those prevention trials possible. Now I'll shut up.
Debating where to go. So, I mean, that was a bit of a teaser. Do you want to follow up on what you guys are working on in the biomarker space that you think is going to help with predicting who will develop which disease when, so that you can design these, so that these clinical prevention trials can be done?
Sure. It's a very challenging question because, as we've talked about, we don't know what we don't know. So we don't know if somebody's going to develop FTD or going to develop ALS. So some standard tests might not apply to everyone. And then that's not good enough in the context of a trial when you're trying to predict whether somebody's going to develop disease or not. So we've been focusing on neuroimaging, which is a little more neutral relative to the phenotype or syndrome that an individual develops. And we think that certain brain regions in particular are more vulnerable than others. And one region is the thalamus that we're very interested in. This projects to cognitive regions of the brain. It projects to motor regions of the brain. And it is one of the earliest loci of dipeptide repeat pathology, which is kind of you can think of it as the precursor to TDP43 pathology in C9orf72. So there's a lot of good biological motivation. We have some good, interesting data that we're about to submit for a paper with Michael, where we've combined the Penn prodromal study that's been going on for about 30 years and his pre-FAL study. And we show that this works across multiple centers as well, and works across individuals who convert to FTD or ALS. It's not a full bullet, though. It helps, and we think that by combining that then with NFL and other markers, we'll be much more successful.
Thank you. So one of the questions that families and individuals at risk often ask is, okay, I want to participate in research, but which studies and how many studies? So for example, there's the pre-FAL studies for people at familial risk of ALS that Dr. Benatar was talking about. There's the University of Pennsylvania studies. There's the ALL-FTD study that we heard earlier today that will, as soon as it's refunded, is going to be recruiting a bunch more people. We are all really excited for that. There's also an all-ALS study that is also enrolling, particularly people with C9orf72 who are at risk of disease in ALS, as well as people with active ALS disease. So if you're from a family that's at risk of both of these diseases, what do you think? Should they try to find the best study and the only study that they participate in? Or if they have the hunger and the willingness, should they try to recruit or participate in more than one study? It's a difficult question, but I think it's one we hear a lot. What would you guys tell them?
I think I'll let Michael answer which study you should participate in, but I'll comment on, as investigators, one thing that we really need to do is talk to everyone running these different studies and make sure that we're collecting similar data that can be pooled together across studies. So if we, again, continue to operate in silos and one study collects this data and it's slightly different from a study that collects other data, this came up in yesterday's conversation about can we combine MRI images from multiple cohorts and different clinicians, and it's actually not a very easy problem to solve. But it can be solved upfront by thinking about the study design. And while each study can have their own unique properties of things that they're focusing on, the more that we work together upfront to make sure that all of that can be pooled together, I think will be important to the success of the mission.
Yeah, so I guess I have a few thoughts, and I don't think there's a right answer, so I'm just sort of sharing ideas. So at the moment, it is the case that studies led by the FTD community tend to focus more on cognition and behavior. And studies led by the ALS community tend to focus more on the motor or the weakness manifestations of disease. So the studies are not sort of identical, and that's problematic. And the investigators as a community have to do a better job of learning from each other so that the studies, in a sense, are more equitable from their approach to phenotyping. Having said that, and while I am amazed at people who are willing to participate, or let me put it this way, be serial participants. Fly here, have a lumbar puncture. Fly here, have a lumbar puncture. Fly here, have 2 hours of cognitive testing, have 3 hours of cognitive testing. I actually think that we're better off studying more different people than the same people over and over. I know from the inception of pre-phals, we've had a waiting list. We struggle to keep up with the volume of interest from the carrier community. And that's interesting because when we first sought funding, the first thing the reviewers said is, the carrier community don't want to do this. And so we said we beg to differ, and I think you guys have all proven that that's true. But it's also the case that participating in multiple studies can confound the data. So when we do cognitive testing, there are learning effects. So if you fly to Miami and participate in our study, and then fly somewhere else 2 months later and participate in another, you learn something and we contaminate the data. So I would actually suggest that you do your due diligence. You pick a study that sort of speaks to you in terms of what the study is trying to do, how it's being conducted, and then push the investigators to share their data across the different studies, to combine data, to use different cohorts for discovery and replication or validation. But I mean, I'm increasingly less a fan of the serial participation because I think it means a smaller subset of the community are participating and we're potentially contaminating data. And so I think we're better off with being in one study. But I could be convinced otherwise, but that's my sort of current thinking.
I think there may also be, I certainly have the impression, I hear from a lot of ALS researchers that they often have a wait list. And all FTD often has a wait list as well. But that there's a lot of FTD studies that don't have a wait list. And so one approach may be that if you see a study that you're really you think that this is something that you could do, that you can help give back to accelerate the pace, make sure you tell the investigator before you ever enroll, look, I'm in another study, do you think that that's going to pose a challenge? Because in some cases it'll be confounding. In other cases it may be asking very different questions. It may be collecting, I can tell you how much just the availability of biosamples is a rate-limiting factor for FTD research. There are so many researchers wanting to do biomarker development, they can't get enough tissue and samples in order to do the type of work needed. That wouldn't necessarily have the type of confounding that you would ask. So I would go a bit half and half with what you're saying, right?
As I say, I don't think there's a clear answer. I see pros and cons. There's also value, which I didn't speak of, is that if we do some assessments on the ALS side, and I will say we're serious about cognitive and behavioral phenotyping, our FTD colleagues might do a better or different job. And then there's value in being able to compare those data. But I think, as you say, it does need to be done in the open, so to speak, that there's clear communications that we can take the best steps to maximize the use of your contribution to the research endeavor and minimize the potential for confounding of the data collecting in both of those studies.
Well said. So we've talked a lot now about the overlap of FTD and ALS. But we also know that there's overlap with other age-related conditions. Dr. Benatar, I was wondering if you could speak about the Miami framework, because that was a really interesting approach to try to unify approaches across neurodegenerative diseases.
Yeah, thanks. I'm happy to talk about it. And this is something that came out of a workshop that we held in Miami, which is how the name came about. But this was an effort to think about studying pre-symptomatic disease, so people at risk for developing ALS, FTD, a movement disorder. And one of the things that sort of emerged from this is that one approach to thinking about this is that we should think about sort of phenotypic streams. So we might think about a motor neuron pathology, so pathology in the motor neuron system. We might think about pathology in the frontotemporal system. We might think about pathology in the movement disorder space or the basal ganglia. These are different sort of neuroanatomies in the brain and in the nervous system. And then within each of those sort of streams or systems, we should think about whether there's no outward clinical manifestation of disease, what we could call clinically silent. If there's a fully manifest syndrome, so in the motor neuron stream, ALS, in the frontotemporal stream, FTD, or whether you're somewhere in between, where there's some subtle manifestations, but not a full-blown syndrome. So in ALS, there might be no weakness, but we see some abnormalities on your reflexes or your muscle tone or your EMG, in which case we might call this prodromal disease. And so we should think about clinically silent, prodromal, fully manifest disease within sort of a motor neuron stream, a frontotemporal stream, a basal ganglia or movement disorder or extrapyramidal stream. And then I guess it's a little bit like not a menu where you're choosing, but it's sort of something from column A, something from column B, something from column C. So you might be silent on the motor neuron, but prodromal on the movement disorders with a little bit of a tremor, and fully manifest with the frontotemporal syndrome having FTD. And we felt this would give us a way to think about this very varied clinical manifestation of this group of disorders in a way that's sort of pure and respects the manifestation and the ability of our ability to phenotype or to understand those manifestations, but to also recognize that there's a shared underlying biology for all of these. And so the idea also is that if there are other phenotypic streams, for example, a muscle stream or a neuropsychiatric stream, that we can add to this as we learn more, and we're not constrained by having to put everything into sort of a single bucket. This might have been more helpful with a visual. I was probably supposed to make a slide and forgot. I apologize. Maybe we can distribute a link to the paper penny. But it was an idea of how we can sort of unify across our field some thinking about these diseases.
And that staging part, I just want to make sure that the reason why is made really clear here, because if we're going to design prevention trials, or if you are at risk of something, does that mean that you should start taking a treatment when you're 20, when you've got 40 or 50 years before you might develop disease? Do you wait until you have almost fully manifest clinical disease? If the disease is starting in your brain 10 years, maybe, before symptoms actually emerge. And that's one of the things that almost all of these neurodegenerative diseases share: Parkinson's disease, Alzheimer's disease, ALS, every form of FTD, they're starting in the brain before the disease manifests. So what this framework is, it's outlining a way to think about all of these diseases in a unified way, recognizing that they're complex with shared features, and a given, say, a C9 family member might be at risk of multiple different phenotypes. But it creates a way to think about how might we run prevention trials as well.
I think that's absolutely right. And it may be, as we're thinking about preventing, say, FTD, that if you have a mild tremor, for example, a prodromal syndrome on a movement disorder axis or stream, maybe that means you're at greater risk of short-term developing FTD, maybe, we don't know that, and would be a very good candidate to be in a prevention trial. And so this approach can help us think about and prepare for this imminent future, we hope, of disease prevention.
Dr. McMillan, I think we were also going to ask you to talk about aspects of pathology, dual pathology that is common across neurodegenerative diseases as well.
Sure. So one thing that we've also learned by looking in a cross-disease brain bank is that everything seems to be connected in some way, which in some ways is surprising. We think clinically of FTD, of ALS, of Parkinson's disease, Alzheimer's disease, but when we look in the brains of older adults, we actually see lots of these pathologies in lots of people. So in Alzheimer's disease, which is caused by tau pathology and amyloid pathology, we now find that about 40% of them also have TDP-43 pathology. So some people claim that given the prevalence of Alzheimer's disease in the population, that Alzheimer's disease is the most frequent TDP-43 proteinopathy. About 30% of those cases also have alpha-synuclein pathology, which is the cause of Parkinson's disease. And in cortical basal degeneration, we see a little bit of TDP-43 pathology, even though we think of it primarily as a tauopathy. So I think one thing that we're learning is that it's also important to think beyond just proteinopathy-related therapies, but to think about common biology across diseases and how that might be informative. One approach that we've grown interested in is VCP, which is a gene that can cause TDP-43 pathology and ALS or FTD, but it can also cause tau pathology. And that was really enlightening when my colleague Eddie Lee made this discovery in our group. But as we work up the biology of VCP, we find that its involvement is related to the clumping of these proteins in the brain. So it's not clearing the clumpiness of all of these proteins aggregating in the brain, which provides a common explanation across all disorders and opens up the opportunity of a treatment that will treat lots of disorders rather than each individual very specific disorder. And time will tell us as we develop these new methods whether the precision medicine approach of dividing everyone into small pieces and having very targeted therapies or a more global disease mechanism approach is going to be successful.
Well said. So we're almost out of time, so I will actually echo what Sharon Denny had said at the end of the previous session, that one of the things we do at AFTD is advocate to have the needs of the FTD space included in all of these different spaces. So we work across the ALS space. We also work in the Alzheimer's spaces. We're really trying to make sure that the larger infrastructures and research that's out there is thinking about FTD in how it's structuring its research. Lots of different partnerships, too many. If we can, where's the slide? Can we get the next slide, please? I don't have the advancer. Okay. Thank you. So any progress in research is made possible because of the community, that we are all part of this together, whether you're able to participate in research yourself or you're able to spread awareness that there are research participation opportunities to others in your community. So opportunities for action, number one, join the FTD Disorders Registry that you've heard about before. There's FTD and ALS resources that AFTD offers, like the FTD ALS checklist, genetic FTD support groups, and resources on symptom management. There are other resources as well for learning more, like the PenFTDcenter.org, ALSresearch.org, and the CREATE Research Consortium that Dr. Benatar has been central to for a long time. They all offer important resources, and they're all on the slide there. There's also an FTD Clinical Research Learning Institute that the University of Pennsylvania is leading with collaboration from AFTD to create a training opportunity for people who are from families who want to learn how do I better advocate in the research space. So it's a training program that has an application basis because the demand has been so high. So the next opportunity will be opening up next year. If this is something that's of interest to you, please stay on the AFTD listserv and we'll make sure that you get that awareness when the opportunity arises. And lastly, please help spread awareness that FTD and FTD-ALS, we're on a spectrum. And it's hard sometimes for communities to understand it. A lot of doctors don't explain it. They may not even understand it themselves because these are complex spaces. So thank you for everything that you do to help be a part of spreading awareness and education throughout your community. And thank you to Dr. Benatar and Dr. McMillan.